Welcome to the open webinar series of IBDC, where we come up every month with open webinar on different IBDC services and data portals to make the research community aware of our services and uh, to outreach how can IBDC and its resources be useful in your research and support your research journey. So I am Sonia Balyan, working as scientist at IBDC, and today we bring to you the third open webinar of IBDC open webinar series. And so this webinar is on Indian Crop Phenome Database at Indian Biological Data Center. So during this webinar, automatically the microphones will be muted, and you can use your chat for asking any questions. The webinar is being recorded for the future reference, and you will find the recording on the IBDC website. So after this webinar also, if you have some queries or you want our assistance in your data submission or any other query or support, you can write to us on support@ibdc.rcb.res.in, or the queries regarding the crop phenome database, you can send it to support@icpd.rcb.res.in. So during this webinar, all your questions were being answered by the ICPD team, including Mayuri Jain, who is the developer in this project, Dr. Nivedita and Ms. Isha. They are the data curators in this project. So they will be answering all your queries during the webinar. So coming to the Indian Crop Phenome Database and coming to the agenda of this webinar, we basically here will learn about the ICPD, its vision, and what exactly it will add to your phenome research and how it can support you in your research journey. We will explore the various features and functionality of ICPD. We will discuss the resources and services provided by ICPD, and also we will just out the services provided by IBDC also. We will give you information about how to submit data, how to access data, and how to get involved with this database and how it can be useful in your research. So by the end of this webinar, I'm sure that you will able to have a little bit understanding about ICPD. You will understand why crop phenome data is essential, and it is very necessary to make it available, and how it can be useful for solving our future problems. You can learn how to submit data, access existing data sets at IBDC, and then you will can also able to discover different resources and support services offered by IBDC and ICPD. And again, you will get to know about contribution of your data and how to you can contribute to the research community and in overall the Indian research ecosystem. So a little bit about IBDC, as we are the just a minute, huh? We are the first national life science data repository in India, and IBDC is supported by the Government of India through the Department of Biotechnology. It enables the implementation of Biotech-Pride Guidelines. It's a joint project of RCB, NII, ICGB, and NIC. This facility is housed at Regional Center for Biotechnology, which is itself is an academic institution established by the Department of Biotechnology, Government of India, with a regional and global partnership synergizing with the programs of UNESCO as a Category 2 center. So coming to what we do at IBDC, briefly I will go through it. And we develop data resources for archiving different types of biological data. We provide high computational facility. We support national projects of importance. We do big data analytics. We provide the users with different resources so that he can seamlessly submit and access data here at IBDC. Very importantly, we conduct routinely different training sessions and open webinars to outreach and to spread the knowledge about FAIR data and the data repositories here at IBDC. So these are the different data repositories at IBDC. For nucleotide data, we have INDACA for crop. We will discuss in detail about crop phenome database. Then we have ISTA, which is the structural data archive portal. We have IPD for proteome data bank. We have image IBIA for image archive, and we have IMDA for metabolomics data. Apart from that, we also support different national projects, and in future we will be coming up with different very useful tools and databases for your research. So coming to the phenome data, what exactly is the crop phenotype data and how it can be useful? So if we follow a proper management approach. So basically crop phenome data refers to any comprehensive collection of phenotype information which is visible, all the characters which is visible of the crop about it encompasses wide range of observable as well as the measurable characteristics. For example, physical attributes, what is the height of plant, leaf size, fruit yield, uh, how tall it is, how many leaves, what exactly is the biomass. Then we have biochemical properties such as nutrient content, metabolic profiles. And then we also look into different various aspects, how a particular environment can affect the phenotype or the response of the crops. So this can involve quantitative as well as qualitative data. It also involves your screening when you screen your wild types versus transgenic at morphophysiological levels. It also involves the small scale to large scale experiments, small scale experiments when you characterize your crops, varieties or transgenic plants in your laboratory or glass houses or culture room conditions, or you grow them at large scale in field trials. So all that data where the phenotyping is involved is again constitute the phenome data. It also involves the high throughput phenomics, as we are having the drone based phenotyping nowadays, which is quite popular and in future will become the most important tool for phenotyping. So why phenotype data is important for India? We all know that India is agricultural powerhouse. We are the second worldwide in farm output, and most importantly, our 50% of the workforce contribute to 20% of the GDP is coming from agriculture. You can see here in this graph, this is the Indian map, and this circle represent the contribution of agriculture in Indian GDP. So why this data is important? When agriculture is important for our country, this phenotype data has the power to bridge the gap between genotype to phenotype, where the studies wherein we want to find out the gene versus trait association, which gene is responsible for which quality trait or which phenotype, so that we better understand how genes control the different important traits. Then the next point is when we focus on improving our crops in bigger breeding programs, we look up for desirable traits such as high yield, better nutritional content, stress tolerance. There also this data is very crucial to not just accurately measure but also preserve and conserve for the future. So then it again responsible for the development of climate resistant crops and data driven insight, so that agricultural we move to manual agriculture to more precision agriculture, where we help the farmers to optimize all the nutrients and water intake in real time, and so that they get the benefit out of that. Then to respond to global challenges, now we have the major challenge is climate change, and we have to ensure for the future food security. So in that case also we need stress tolerant crops, we need better yield crops, so that they can support our food security. Then for many of our comparative studies where we compare different crops in different environments, so that we can figure out which is the variety is uh, selected for a particular environmental condition or a geographical location. Then it is the foundation for all our multi omics integration, where we're integrating genome, transcriptome, phenome, and these information to chunk out the holistic view of the crop development and adaptation. So in a way, this crop phenotype data is very important, where we've for the future questions regarding food security, precision agriculture, and data-driven decision making. So, but what exactly are the challenges with this data? You know that in India we can experience in laboratories. We are carrying out these small scale phenotyping experiments where we are self measure. Then in fields we have a workforce who measure. Then we have certain software who measures the phenotype data is also influenced greatly by environment, then the genotype, and then the all those methodologies. And these methodologies are not standard, so we don't have standard protocols, we don't have standard machinery or data standards to record that observation, and these again vary from crop to crop. So the overall the phenotype data is very heterogeneous, and itself it is reflecting that all the experiment goal design setting, agronomic management, it varies a lot, so making it very complex, so that it is get in assembled by humans and machine. And due to the lack of standard or international repositories, it is very poorly documented. So usually a researcher carry out whether it's he carried out small experiments or big screens of the varieties, but this data is always published and go into some figures and never can be traced back in future to reuse, repurpose to generate new information. So the major problem is the difficulty of reuse, because we don't have right now the standard repositories for this type of data, uh, unlike we have for all the omics data. So this is a greater challenge in making this data fair in future. So what we have to do, we have to follow proper data management practice, so that we can ensure that our research output become knowledge and not trash. So here is the a standard data cycle. It is relevant for every and each type of data, but you can imagine it as a phenotype data where you plan your study, then you generate a lot of data with a lot of your hard work, then you collect that data, process that data, analyze and publish your research, and then you forget. And it is never going to be reused, repurposed to generate new information. On the other hand, we have a fair data management practice, where this data after the publication is preserved properly, shared in a repositories with valid accessions, with valid use of standard vocabularies, and can be reused to generate new knowledge to solve the future challenges. So with this in mind, at IBDC, we have worked to develop this Indian Crop Phenome Database, so that this type of data can be archived in a fair manner. So the vision with ICPD here, we have that we have to create, or we wanted to create a centralized user-friendly platform to digitize, disseminate of the Indian crop phenome data aligned with the fair data principles. And thereby, to achieve this vision, we have to, or we are doing structured metadata fields and ontologies to enhance its findability, interoperability, reusability of this data, so that it will empower the research and development. So the bigger picture here is by adopting these data practices, preserving and submitting data to proper data repositories, we can achieve that in a way in future, we can achieve that these type of portals or ICPD can be able to select stress tolerance cultivar. We are able to select crop based on the geographical location. We can select trait of our interest. We can understand the genome phenome interactions better. We can develop resources for further empowering the breeding and crop research. So coming to the inspiration of this database, we have followed the international standards, and here is the minimum information about plant phenotyping experiment MyAPI. These are the checklist or the main information or the checklist data standard for archiving the phenotypic information. So we have considered both MyAPI and GNPIS for the development of Indian Crop Phenome Database. And you can imagine that ICPD takes care whether your project is a single user project. Imagine that you are a PI, you have a project and one crop, several studies, several data files easily managed through ICPD. Or you have collaborations with different PIs, you work on a single project, or you work on a multiple project with multiple crops, or single crop, multiple studies, and multiple data files. So irrespective of whether your phenotypic is carried out in field, greenhouse, large scale versus small scale, wild type versus transgenic, you have recorded multiple traits, morphological to physiological traits, any type of data can be submitted at ICPD. So with this version 1, whenever a study is submitted here, the data submitted in ICPD will get a project accession, a valid persistent identifier, a study accession, and the data file accessions. Here we are using controlled vocabularies, and whole this process of data submission is ontology enabled, and we use ontology terms for trait, tissue, developmental stage, and methods, so that data can be interoperable and easily archived. So this is the journal overview of the process here at ICPD. Any user who wants to submit data can register into the portal, then submit. He can register after the authentication. He can log in into into the ICPD dashboard, and after landing onto the dashboard page, there are many options to submit new data or playing around with already submitted data. So the first step is project registration. The second step is study registration, and the third is generate the template and add your data and upload. So the process is very simple, and after uploading, you can access that data. So any researcher can access the data submitted at IBDC, and we have two modes: open access and controlled access. So in open access, the data is straightforward, open for public, and in another you can restrict your data up to 2 years, and you can easily download all the metadata related to study, project, and even the data files can be downloaded by the users. So coming to the controlled vocabularies that we are using here in this process, we are using ontology terms from crop ontology and plantiome, so that we can minimize the redundancy while recording data. So we will using ontologies at developmental stage, tissue, traits, and methods, and you can submit any crop data at ICPD. So in case if your ontology terms were not there, we have two functionalities where you can browse the ontologies of your interest, and you can see whether it is there or not. If it is not there, you can simply create the ontology, so that in a way you can also contributing towards building new ontology terms, and it will be assigned a temporary ontology accessions, which later will be submitted and curated and assigned a permanent accession. So coming to the metadata model that we have followed for this portal, we have three basic divisions. One is the project, where we require information about title, grant number, the funding agency, the project description, associated publications, release date, collaborations, and crops. The study requires details or metadata to be filled related to basic details of the trait, study, description, start and end date, location of the experiment, experimental design, field design, growth facility, tolerant and susceptible checks if used, growth and environmental conditions, temperature, light, humidity, or any other condition, what type of treatments are used, whether there are no treatment, biotic or abiotic treatment, what agent is used, what exactly treatment, how you have given the treatment, what is the qualifier, duration, developmental stage. All these metadata are required, and it will characterize your study to a level based details, so that you can anyone who is using your data in future will have all the information, so that easily understand your data. Then we require a most important section is the trait, how many traits you have recorded, what tissue, what method, and finally who all contributed to the study. Then after that we have to generate the template, list the accessions, and select out the technical and biological repeat, and good to go. So this is the ICPD dashboard. It looks like this is your dashboard. So when you log in into the portal, you will see this dashboard where you can have options for managing your data. You can see what all projects you have already submitted within accession. Please pardon us because it is our testing screenshot, so you will file these descriptions. So you can also see whatever you can see and download all the metadata related to project as well as study. You can see these files, data files, and download it, share it with anybody. Then you can give your feedback if you encounter some suggestions or you have some technical support required. Browse and create ontology sections so that you can manage whatever ontology you want to use. We have a manual section where all the detailed protocols are listed. So this is your go-to dashboard where you can easily manage all your phenotypic data. So coming to step one, how you have to start with the submission. The first step is the register pheno project. When you click onto this section, you will find this form where you have to add the title, grant number, funding agency, project description, associated publication, if any. You have to set release date, and release date cannot be more than 2 years. Then you can also select whether your project is individual or in consortium. So if it is in consortium, you have to fill out the details of other PIs. You can add multiple PIs depending upon your project. Then you have to select crops here also, because nowadays we are getting bigger and bigger project where multiple crops can be there. So you can add one to many crops within a single project. So after that, when you successfully submitted the project, you will get an project accession through mail and also displayed on your dashboard. Then the second step is the study registration. You can register multiple studies within a project. For study, you have to select your project, select the crop in which you have done the study. Then you have to add meta trait. Meta trait is basically a bigger trait. For example, you are screening your cultivars for drought resistance. But for drought resistant, you are further uh, studying a leaf rolling, or you also be dealing with the water efficiency. So these are the trait, and the drought resistance is your meta trait. So then you have to filled out the study title, description, start and end date, location, growth facility, susceptible and tolerant check if you have, the information about growth and environmental conditions. Very simple form is this. Then you have to select out what all treatment you have given, whether it's treatments or no treatment, biotic or abiotic, for how much duration, what development stage you have started your treatment. So it will allows you to add single, or if you are working on dual stresses, you can add multiple treatments also. Then multiple traits can be added. Any number of traits can be added. You type here, when you type here, automatically the ontology terms or options will be shown. You have to select one based on your best term, and if the term is not there, you can also create it. Then in the last section, you have to fill out the author details and you are good to go. So after the successful submission, you will get an accession for your study. And then now the third step is the generate template. So whatever metadata you have recorded, you have to select your study and list your accession list is basically the name. For example, if you have 1,000 accession, so 1,000 accession list, or the name of cultivars, or the name of varieties, genotypes, wild type or transgenic, whatever names, then you have to select out how many biological and technical replicates were there in your study. So this type of a template will be generated, which will have pre-filled fields, and you only have to fill this section of the observations. So after filling the observation, you just select your study and upload the file. This is a simple CSV file, and it can include n number of cultivars, n number of traits, depending upon your study. So after the complete submission, you will get accessions for your project, you will get accessions for your study, you will get accessions for your data. And after the submission, automatically the project study will updated onto your my data page, and you can easily download all the metadata, share it with anybody. You can easily see what all information is there in the study. You can see what all files you have submitted. So this is the data statistic. As of now, we have 16 successfully completed studies there, and these studies will, again, we have 21 developmental stage, 16 traits, 18 tissues, and more than 500 cultivars. And but all the submissions are from one organism, that is rice, and all of the data is open except for one study. So you can check out at the browse data page of ICPD. You will find all the studies that are already submitted. So whatever studies are controlled access, you will find the download link, and the studies which is under controlled access, you will find a non-highlighted link here. So for all the open access data, you can easily download the files and reuse for generating new information. So in this way, you can make this phenotype data fair, so that anybody in the world can able to find it, access it, and reuse it for generating new information, so that you will also get the more citations. Your work will get more outreach or visibility. You will get the uh, the uh, um, the credit for whatever hard work you have done, and the data can be repurposed to generate new informations. So in future, we definitely working on the next version of ICPD, where we again want to integrate or associate the phenotype data with the other type of data that we have at IBDC, so that we in future can able to generate crop specific data resources. So here is the team at IBDC. This is our IBDC team. These are the PIs, Professor Arvind Sahu, Dr. Debashish Mohanty, Dr. Dinesh Gupta, and Professor Deepak Nair, who is the acting head of IBDC. We also acknowledge the support from Department of Biotechnology, IT-RCB, and Professor Raghuvanshi, who is our former read. This is the team ICPD, and special thanks to Vipul and Rajesh for the assistance in organizing this webinar. I hope you will able to have a little bit about information about the how to go about the submission of the crop phenotype data. So in case if you have any questions, we can happy to answer. Or if no questions were there, you can also write to us at the support, and we will able to help you out with your submissions also. So I hope this will be useful, and you will now able to submit data to Crop Phenome Database. And thank you again for joining us today. And I think we don't have questions. So I think we can stop here if no questions were there. So thank you for your patient listening, and we look forward to our upcoming webinar on Indian Structural Data Archive and in the coming month. So do join us for that also, and thank you so much for joining us today.